Good morning, USA, and good evening, India. I am Dr. Ritika Shah, and I would like to welcome you all to the GCAR Journal Club July meeting. Journal Club is an educational initiative by GCAR where we discuss published research articles and critical analysis to stay updated with latest research and promote evidence based Ayurveda. GCAR, or Global Council of Ayurveda Research, is an international non-profit organization based out of USA with a sister chapter in India by the name of Ayurvidya Anusandhan Abhiyan Foundation. Our mission is to encourage, educate, and facilitate basic clinical and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence based health science globally. You can learn more about us at www.ayurvedaresearchusa.org. Let me quickly share the website with you so you can have a look with the foundation and the aims and goals of our nonprofit organization. Mm, This is our website. You can go over our website. Please consider us joining as a member, which will bring you access to all our resources. And no nonprofit can operate and succeed without our volunteers. So consider joining us as a volunteer, and we can place you in a variety of projects based on your skills and interest. Once again, our website is www.ayurvedaresearchusa.org. Now, um, let me stop sharing. Now, I would like you to introduce our eminent speaker for today, Dr. Swati Ramamurthy. Dr. Swati is an MD in Ayurveda. She started her career in the herbal pharmaceutical industry right after her post graduation in 2018 as a formulation scientist. Later on, she moved to Central Ayurveda Research Institute, an apex body under Ministry of Ayush, to work as a senior research fellow. She was handling the tribal healthcare project during her tenure there. During the pandemic, she was actively involved in three COVID research projects and was single handedly responsible for executing it on the field. The number of subjects in each research project ranged between 800 to 1500. Later on, in 2021 December, she moved to Herbal Chakra Private Limited as head research and product development. And since then, she has successfully collaborated with the team in house as well as various labs in Delhi and Noida and launched around 75 new Ayurvedic products, driving significant growth and profitability for the company. Her focus to attention to detail and organization skills have enabled her team to develop innovative formulations and ensure the highest standards of efficacy and safety. Her Along with this, her other achievements are she stood seventh in state RGUHS for Department of Rasashastra and Bhaishajya Kalpana in MD. She was awarded Best Oral Paper Presentation for role of Bhasmas in management of diabetes at NEMACON. She was also awarded Kalasri for overall achievement in cultural activities and and Ayur Visharda by Himalaya Drug Company in 2013. And she was also awarded Pratibha Puraskar by Bubur Kame Seva Samiti for securing seventh rank in MD. She has given several talks and seminars in and spoke in several conferences. There is much more, but I think for now it's enough. Let we will let Dr. Swati speak for now. Over to you, Dr. Swati.
Thank you, Dr. Ritika. First of all, thanks for having me. Thanks for this opportunity to be able to present and to be able to interact with the community and help in scientific growth. So I'll start with the topic directly. Today, I have chosen the paper. Just a second, let me share my screen. So today's topic would be new approaches to breast cancer therapy through green nanotechnology and nano Ayurvedic medicine, preclinical and pilot human clinical investigations. So there are a lot of reasons I've chosen this paper. I'll come to it eventually when we start discussing this in detail. I would just like to highlight a few things here when we look into the topic. It's mainly that one is cancer research. Next is nanotechnology, green nanotechnology. That's what most of our bhasmas are about, because we use plant material, phytochemicals to make sure the bhasmas attain the nanoparticle status. And because of that, I wanted to focus on this one because bhasmas are generally studied for what they are, and we don't have much of uh, evidence on the fact that how bhasmas really work. Most of the things that we have already, published, we find in publications are mostly about, hypothesis as to what happens inside a bhasma, how the particle changes and all. But we don't really have a proper complete study when it comes to preclinical trials and then clinical trials and all. So that's one of the reasons I chose this paper, because it's completely, it starts from preparation and characterization of the medicine and ends at pilot study. So they have taken us through the entire journey of a production of medicine and then seeing how it works and then proving its efficacy as well as safety. That's one of the reasons I have taken this. So this a bit of details about the publication and when it was done. So this was published in International Journal of Nanotechnology. Again, the relevance part is very important here because these are the journals which reach out to a large population. I mean, if it is something related to Ayurveda journals, what happens is most probably Ayurveda people might go and have an access to this, go and read this. But when it comes to people from different fields getting a insight into what nanoparticles are, what bhasmas are, and how we can, have a collaboration to improve the product or understand its functionalities and all. Other people don't get exposure because of the because of the restriction when we publish things in Ayurveda journals. So by publishing it in a journal of nanotechnology, which is just the core field, this helps in providing access to a lot of people where people might be interested in taking up further studies on these things. This article was published on Jan 13th, 2020, pretty recent that way. And as you can see, there are like more than 6 authors for this, and most of them have worked in University of Missouri, Columbia. So you can see that Department of Radiology is involved for the investigations and all. Institute of Green Nanotechnology is involved for the production of the medicine. Also, Dhanvantari Nano Ayurveda Aushti, Dhanvantari Nano Aushti Private Limited is a pharmaceutical company, so they were sponsoring the study. And Department of Physics, Department of Pharmacology, Department of Biological Engineering, all these departments were involved. Hence, this makes the article more interesting because there's a perspective from every department involved here. So we have the Green Nanotechnology department where they're focused on green nanotechnology and production of nanoparticles using phytochemicals. And that we can say is a more advanced a more advanced way of looking into bhasmas. Um, and then we have Department of Radiology for all the analytics and all. And then we have Department of Physics, Department of Pharmacology for all these animal studies. And then they even tied up with a organization in India to conduct some pilot clinical trials in as per Ayush regulations. We'll come to that later. So this has a lot of people working who are from different fields, and they give their input as to how a product can work, or how the characterization of Swarna Bhasma for that matter. All those things make a huge difference in understanding the pharmacodynamics and pharmacokinetics of the product. And then when we come to impact factor, the first quarter in 2023, their impact factor was 6.6, and 5-year impact factor has been 7.5, and they're indexed in PubMed Central, Scopus, and Embase. And before we begin with this presentation, I want to just clarify one thing, and with this disclaimer that this presentation is for academic purpose, and we are doing this to understand and learn how to present well, how to write good research studies, how to conduct good research studies, and how to publish good research studies. So it's an open discussion, and the critical appraisal is also for that only. Excuse me. Just a second, I'm a bit under the weather. Just give me a minute.
No problem, Dr. Swati. Take your time.
Yes. So whatever we discuss here, whatever we look into from a critical perspective, is mainly to understand a study better, mainly to see how much well we can do further when we are publishing, or to understand more about the intricacies of publication. So this is not intended to slam anybody, or criticize any publication or anything of that sort. This is just for more of a growth and development as a researcher, and it is for academic purpose. So with that disclaimer out of the way, let's start with the study as such. So if we look into the paper, first part, like we'll be following the IMARD format, that is introduction, methods, and then results and discussion. And then finally, I come to the aspect about critical review of this paper. Excuse me, this will keep happening. I'm sorry. So yeah, first, when we see the aim of this paper, so they state that the main aim is to investigate the potential of green nanotechnology to modernize Ayurvedic medicine for breast cancer treatment. So when we look into this, green nanotechnology, let's understand a bit about what green nanotechnology exactly is. Green nanotechnology is a subfield of nanotechnology where they use phytochemicals to develop nanoparticles. So they have various methods through which they encourage an interaction between the phytoconstituents and metal salts to generate nanoparticles. And when we look into it, although the word seems fancy and all, but if we go back a few thousand years and look into Rashastra, that's what we used to do when it came to usage of bhasmas. So bhasmas are generally byproducts of interaction between the metals, minerals, ores, and phytoconstituents. And that's the same way green nanotechnology also works, although where bhasmas are involved, there's a lot of concept around heating it in particular temperature using putas and all. Whereas in green nanotechnology, they're taking that out of the equation, and instead they're just interacting the salts of the metals with herbal products like phytoconstituents. And based on that, so this basically in this paper, they have derived the inspiration from the concept of bhasmas, and then prepared a medicine using green nanotechnology. And then that has been tried in the management of breast cancer. And most of this has been tried in triple negative breast cancer. So that's one of the more aggressive forms of breast cancer. And right now, whatever treatment modalities we have in management of breast cancer, although they're pretty established and they're working really well, there are a lot of side effects associated with those treatments. And sometimes the side effects outweigh the benefits. Because of that, we need better interventions for these conditions, and especially cancer treatment has been the biggest challenge in today's society, considering all the side effects and the negative effects it brings in. So, bringing out safer alternatives for cancer therapy um, is what I can call the need of the hour. And that's why this research is very much apt for the current times. So, the objectives, they describe the objectives in a very clear cut way. So, first one is to characterize the gold particle based nanoswarna bhasma. Second one is, it's abbreviated as NSB. Second one is evaluating its in vitro anti-tumor properties, that is, cell line studies have been done, and then preclinical studies to understand the efficacy in animal model, and then pilot human study for metastatic breast cancer. So they go by the research hypothesis that nanoswarnabhasma drug, a combination of gold nanoparticles and Ayurvedic phytochemicals, will demonstrate significant anti-tumor properties and be effective in treating breast cancer with minimal toxicity. So they're going in with the idea that they have to assess the efficacy, safety, and then efficacy. So safety is the primary issue concern here, and then next is the efficacy. So when we look into methodology, they have in this paper, what they have done is they haven't gone for a separate, character synthesis and characterization paper, and then a separate cell line study paper, and then an animal study paper, and then a pilot study paper. What they have done is they have integrated all these studies into one paper, and that is helpful because that's something I really liked about this paper as well. Because generally, what happens is it's a little hard to find, the continuity paper. For example, if someone publishes a synthesis and characterization and then they go for the saline study, so from there to the next paper, it's hard for a researcher to keep finding it, even if it's on PubMed or any platforms. It's just, hard to keep the continuity of it, maintain the continuity. So when you have everything in one single paper, it's easy to refer to and easy to understand, and give a sequence to it, because of which at least the all the evidence is put together in one paper. That becomes very easy. So in the methodology, what they have done is initially they have synthesized and characterized the gold nanoparticles, and then they have gone for preclinical studies. In the preclinical studies, they have cell line study as well as the animal study. In the cell line study, what they have done is they have used the MDAMB 231, that is the triple negative breast carcinoma cell lines, and then the human aortic endothelial cells, that's the normal cells. So both of them they have used to assess the cytotoxicity and apoptosis of effect of nanoswarnabhasma. And then when it comes to animal study, what they have done is they have used SCID mice inoculated with tumor cells, and then they have made it into three groups. So group one is the untreated control. Group two is getting the NSD drug in a dosage of 3 milligram per 30 grams of body weight, whereas group 3 received 7 milligrams per 30 grams of body weight. And then they were assessed for tumor volume, body weight, and overall health. And then ultimately, animals were sacrificed at the end of the study. Next, they went to pilot study. So in pilot study, they have selected only 6 patients between the ages of 18 to 65, already diagnosed with stage 3 or 3B breast cancer. And then they were divided into 2 groups of 3 people each. Randomization was done. And then there they have given standard care for one group, and the other group standard care plus NSB drug. So, and then they were assessed with quality of life questionnaires, and then CT scan with racist criteria, and then NCI toxicity criteria, because they are also evaluating the safety of the drug. Toxicity and adverse drug reactions were evaluated or looked into. So when we see say standard care and standard care plus NSB drug, the standard care involves doxorubicin injection 60 mg per square meter, 3 weeks as 1 hour infusion, once every 3 weeks as 1 hour infusion, plus cyclophosphamide 600 mg per meter square, 3 weeks again. And when it comes to standard care plus NSB drug, along with the standard care, NSB capsules, 2 capsules thrice daily after food every day. So this was the treatment routine that was implemented. Now, when we look into NSP preparation and characterization, so this is important. I have taken a separate slide to highlight this because they have mentioned how they went along with the process of preparation of this drug. And they have clearly mentioned in the paper as well that they have tested it for scalability. That is one very important thing because when we study most of the drugs, we develop the drugs at a research lab scale, and then we study its safety and efficacy. And while we are doing all of this, most of the times we tend to forget how to scale this product, how to scale this up and bring it to public domain, because ultimately when it goes into public and starts getting used is when uh, you know it it's its ultimate goal or ultimate purpose is served. And to be able to do that, scalability is very important. And most of our medicines, when we try it in the lab scale and then when we go for production, the scalability becomes a huge problem. Even when I'm speaking about basmikarana process, it's a very tedious process. And to get 1 kg of basma or something, it takes a lot of labor as well as a lot of resources in terms of electricity, fire, and then phytochemicals, the processes, the time, everything. So scalability is very important. And here what they have done is they have prepared it, they have characterized it, and then they have made sure that this is scalable for production as well. That's one of the best parts about it. So how they have went about this is they have taken the reference from Ayurveda for usage of amra in manufacturing Swarna Bhasma. So based on that, they have isolated magniferin, and magniferin is an active component in the mango peel. So they have taken that, and with mango peel extract, which contains magniferin, they have added the um, auric chloride, sodium auric chloride. This one, it's a gold salt. Basically, they have mixed both of them to generate AUNPs, so gold nanoparticles with magniferin which is surrounding them. Then they have gone on to characterize it using TEM. It's a type of transmission electron microscopy, where particle size assessment as well as what is the shape of the particle, what is the dispersion of the particle. All of these can be studied. And based on characterization, then what they have done is to those gold particles, gold nanoparticles, they have added amla, haridra, that is curcuma longa or turmeric, glycyrrhiza glabra, mulethi or eshtimadu, acacia. So all these things they have added to it based on again the Ayurvedic principles of bhasma manufacturing, where we use all these herbs in preparation of Swarna Bhasma. We find various references in various texts about manufacturing Swarna Bhasma using these ingredients. So they have added it with that one. And then they have produced the final NSB drugs. So next, when we come to results, so I'm not discussing the next parts of methodology because it was there in the methodology slide itself, where I was talking about how the preclinical and clinical studies were done. So when we come to the result part, in the characterization of NSB, so characterization of NSB as in after preparation of Swarna Bhasma or nano Swarna Bhasma, they had to look into the details of that particle. Like, they had to characterize it in terms of what is the particle size, what is the shape of the particle, what is the zeta potential, because depending on the zeta potential, there's agglutination or, piling up of the molecules that has to be avoided. So there has to be some zeta potential and all, so that it's easy to suspend them in a colloidal state. So all these things have to be studied, and these are also an indicator of how effective they can be when they go into body, how much they can be absorbed, and how much they can be excreted in a safe way. So that characterization was done, and also they studied for what is the quantity of gold that is present in the NSB drug. So the production of MP, AUNPs and NGF, AUNP. So yeah, here MP, AUNP is the mango peel, gold nanoparticles, and MGF, AUNPs is magniferin, gold particles. So what they have done is they have taken mango peel extract, and with that they have reacted the gold salt, and they have also done it with direct magniferin extract. So they have taken, magniferin they have taken from the mango peel extract, and then they have reacted it with gold particle also for comparison. And then they have confirmed that these two particles exist. Then in the preclinical studies, in the in vitro as well as in vivo, that is in vitro is the cell line study, they have seen that NSP drug showed a dose-dependent reduction in cancer cell viability with minimal effects on normal cells. So why is this important? Like, why minimal effect on normal cells is important? I think most of us as doctors are aware that most of the chemotherapeutic agents, although they have been told to be selective when it comes to cancer cells, they do have an impact on the surrounding normal cells as well. And they do have a huge impact on other parts of the body in terms of immune system and all that. Because of which, we need an effective medication which doesn't touch the normal cells but is very, very selective to the cancer cells. That's where this finding becomes very significant. And then when we see it in the animal studies, significant reduction in the tumor volume in NSP treated mice compared to controls. So if you can remember, there were three groups. The first group of mice was untreated control, and then the second and third were 3 grams and 7 grams, 3 milligrams and 7 milligrams dosage. So they had a significant reduction in tumor volume as compared to the controls. Next, when we come to clinical trials. So this state that in the clinical trials, their primary objective is to understand the safety, and the secondary objective is efficacy. So the majority of adverse events were mild, and one severe adverse event, but it was unrelated to NSP drug. So that severe adverse event actually happened in the standard care arm, not in the standard care plus NSP arm. And then in the efficacy part, you can see that NSP drug plus standard care group showed 100% clinical benefit rate. So I'll come to the details of that one, what exactly is a clinical benefit rate and all. And then there was partial response, that is 66.7% of patients in the standard care group and 33% in the NSP group. And then stable disease in 66.7% of NSB group patients with no progressive disease. So in the three people who were given NSP drug plus standard care, the disease was stable with no progress. So the cancer did not progress and it was stable. It did not start metastatizing or something of that sort. And that's a huge win when it comes to cancer care. Now, if we look into the characterization of NSPs, so even if you go through the journal, published paper, you can see that they have clearly discussed about how the particle looks, what should be the particular wavelength, what should be the UV visible spectrum characterization, and then TEM characterization, and then STEM images are being given, to make sure that even when this is scaled to a commercial level, to an industrial level, even then, if these things can be maintained, then you have a standardized product. So that's one of the very important things, because standardization is something we face a huge challenge when it comes to Ayurveda. And especially when we speak about manufacturing bhasmas in the traditional way Ayurveda has told, going with those temperatures and then going with those, phytoconstituents to make those bhasmas, even a minor variation in the quality of the herbal drug we are using to manufacture bhasma can lead to a lot of changes in the end product. So standardization becomes very important. And here they have given a clear standard as to these are the parameters to which it should fit in, and that will make it viable, and that is the standard for it. So that's one of the best parts. And if we look into the physicochemical data for the parameters, You can see the core size is in nanometer, and then what is the absorbance, and then hydrodynamic size, zeta potential, and then how much gold in gold nanoparticles is present. So they have done it through atomic absorption spectrometry. And you can see that with magniferin extract or mango peel, both of them, the parts per million of gold is similar. Like, it's almost the same, 327 and 327 parts per million. Next, when we come to the cell line studies. So as I was saying, they have used two types of cell line. One is a normal cell line, and the other one is the MDAMB 231 cancer cells. So here is the concentration, micrograms per ml, and we can see that cell viability percentage of control. And this is a 48-hour graph where you can see that in the cancer cells, there is a eventual drop in the cell viability. So this proves that the NSB drug was effective in preventing, inducing cytotoxicity in the cancer cells. And then when we go to preclinical studies, so what they had done is they have taken SCID female mice, and then they have injected the tumor cells into them, and they have after a certain stipulated time when the tumor started growing up, that's when they start treating them with the medicine as well as control group in the untreated way. So they have taken 7 animals in each group, and treatment was given twice per week, and animals were randomized and treated orally on day 0. So their day 0 was the day of randomization and starting the treatment, not from the time the tumor was induced. Then in the untreated control group, only tumor-bearing SCID mice were monitored. So they were monitored for their tumor size, their body weight, and then overall wellness. Then it is very significant to understand that NSP significantly inhibited tumor growth compared to control. So the significance confidence interval they have taken is less than 0.05. P is, and then reduction in the tumor volumes by over 80% during 60 days of treatment. So they were being treated for 60 days, and the reduction in tumor volumes was by 80%. That's very huge number. No significant body weight loss or other signs of toxicity were observed. This becomes very important, especially in reference to cancer treatment, because most of the chemotherapeutic agents that are used in cancer do have a negative impact in terms of weight loss, nausea, and then disturbances to other gastrointestinal functions. And this proving that there's no significant body weight loss in the animal model is a huge win. Next, when we come to the pilot study. So when we are speaking about the pilot study, it's important to understand that they have clearly mentioned it is a pilot study. So one of the primary concerns here is in the pilot study, the number of subjects being enrolled are very less. So they have only taken 6 subjects, of which 3 are in the treatment group, that is the arm A, and 3 are in the arm B. So arm A consists of standardized treatment, and then arm B is standardized treatment with NSP drug. So total, they have taken only 6 subjects here. That's a very small population to study. And yeah, although they are calling it a pilot study, that's something which is which has to be probably studied further. That is one thing I found lacking in this paper, because just 6 people, even for a safety study, is a small population. Especially considering the incidence rate of breast cancer, it is a very, very small population, and it's not inclusive population. Because of, I think, so that's one of the limitations of the study as well. And now that we have discuss this, let's just look into a brief understanding of what exactly went to, what were the results when it comes to the tumor response. So no one had a complete response. So complete resolvement did not happen because, of course, the treatment was done only for 12 weeks. So the summary of overall target and non-target lesion response at the week 12. So they have done only 12-week analysis. So complete response was not seen in either groups. Partial response was seen in arm A and arm B. One person showed partial response. And then stable disease. So in the arm A, the stable disease was 0, whereas in the arm B, that is the treatment group with NSB, the stable disease was 66.7%. 7%. And in arm A, 1% showed progression in spite of treatment, whereas in arm B, there was no progression that was noted. And then when we speak about target lesions, there was no complete response in the lesion, and partial response, again 2 and 1, and stable disease, that is the lesion did not grow or increase in size, it remained stable, was 0, whereas in arm B, that is the treatment group, it was 2. Progressive disease, uh, the disease progressed in 1% in arm A, whereas it did not in arm B. And then non-target lesions, there was no metastasis involved. So non-target lesions was 0 everywhere. They have taken it as baseline. And it's important to understand that disease control rate or clinical benefit rate, as we call it, like, the benefit from the medication was 100% in arm B. And there's also a waterfall plot. So waterfall plot is a common thing that is used in cancer studies because it considers a lot of parameters and it can individually depict the subject. This is very useful when the subjects are less in number, when it's a very small sample size. And then when we come to the discussion part, they have discussed mainly the pilot study part in detail, whereas the efficacy and animal study have not been really discussed in detail. Safety and efficacy in terms of animal studies and cell line studies, but they have focused their discussion more on the pilot study part. So in the discussion, some of the few main points that they have mentioned is in doses in the range of 3 to 7 mg per 30 grams of mouse administered 2 times a week are able to control the growth and proliferation of tumor in a highly effective way as compared to the untreated control groups. Efficacy was a secondary objective. Mean sum of the longest diameter of target lesion in treatment arm B, standard care treatment, was reduced at week 7 and further reduced at week 12 compared with the baseline measurements. And then 2 patients in treatment arm A and 1 in treatment arm B showed partial response. 2 patients showed no change in the lesion, that is stable disease in treatment arm B, and then progressive disease response was observed in 1 patient in the treatment E. None of the patients in treatment arm B showed progressive disease. Yeah. So now I'll come to the critical review of this paper. And initially I told a few points as to why I chose this paper. I'd just like to highlight a few more things about it. First one is, when it comes to Rasashastra specifically, although most of the Ayurveda drugs, yes, but still the drugs of Rasashastra, we lack standardization. And if we look into some of the most popular pharmaceutical companies that are manufacturing Rasaushadhis in India, um, majority of them have their own standards and they manufacture according to those standards, and that too only for popular bhasmas, like it might be a lauha bhasma or a popular abraka bhasma, yashoda bhasma, but most of the times there's a lack of clarity on standardization, and most of the times there's a lack of clarity on characterization, on the temperature controls while manufacturing the product. And although we have lab scale standards, they haven't been proven to be this, efficient or effective when it comes to scaling them up for a industrial level. Because of this, what happens is there's a, there's the efficacy of the product is hampered. There are safety considerations that have to be taken care of. And because of all these things, people are very apprehensive about writing um, Rasaushadis and consuming Rasaushadis as well. And we have seen instances of Rasaushadis being toxic in a lot of cases because of improper manufacturing or poor standard quality control standards. So it's very important to characterize nanoparticles, standardize them, and this paper addresses that issue. Although we might not directly go with the, excuse me. Although we might not directly go and use the green nanotechnology per se, excuse me, we have a lot of methods from this to be taken and implemented in standardizing our bhasmas as well, right from using a TEM to AS and characterizing bhasmas. We can do a lot of things when it comes to bhasma study. So this paper can be a very good example of how we can try and standardize the herbometallic preparations or herbomineral preparations of Ayurveda. That's one of the reasons I chose this study. The second part was this paper, one single paper has the entire product cycle. So it's not just one aspect of research. It starts from development of the medicine to the uh, safety and efficacy of the medicine. So it gives a detailed account of what was done in all these things. And the next important part is, although they have gone through the details of all these methods, like they have included the preparation of and characterization, and then they have gone through the preclinical and then the clinical, they have made sure they have clearly explained the methods for all of these. For example, if we speak about characterization and preparation and characterization, they have given a clear picture of how it is prepared, how it is characterized, what are the measures that were taken to characterize it, and then they go ahead and speak about how the cell lines were obtained and what was the exact study done on the cell line and what was the exact result that it gave. And then they go into the randomization of the mice and then inoculation of tumor, how many days they waited for the tumor to grow properly, and then when the randomization was done, how the medicine was given, and then finally what time the animals were sacrificed, what all were studied for. Everything is in detail. Although every 3, 4, 5 different studies have been included, everything has been given in pretty good detail, and it's a very clear cut. That's one of the reasons I selected this paper, because this is one of the examples of how good a paper can be written. Um, and then the another important part was cancer research. We have a lot of anecdotal evidence. Even today, there are a few popular practitioners in India, especially, who claim to treat cancer based on by using rasaushadis or proper Ayurveda, herbal medicines as well. They claim to have treated cancer, but most of them are anecdotal evidences. We don't have empirical research-based evidence that this herb or this formulation or this combination works effectively in cancer. And because of that, what happens is it becomes hard to implement those medicines in actual cancer therapy. And considering the current prevalence of cancer, I'm not just talking about breast cancer here. When I speak about cancer, it's cancer in general. Um, the prevalence is very high right now. I mean, if I look into my own circle, I think at any given point in time, at least 1 or 2 people in my closest family have been going through cancer treatments, or some of them are in remission. It has been that common, and I have lost a few people for cancer as well. So cancer is very, very prevalent these days because of a lot of reasons which are in our control and which are not in our control, like environment, the toxins that we consume on a day-to-day basis, plastics. There are 1,000 reasons that are giving us cancer. But the medicinal options to that are very limited, and most of the times it's either that you choose to live with a poor quality of life or you choose to die. That's the poor status of cancer management even today, in spite of so much of advancement in medicine. So integrated approach, when it can improve the quality of life, when it can actually help in addressing a lot of issues, a lot of side effects that comes along with cancer, and also can act effectively in managing cancer without giving that kind of a toxicity or negative impact. Those things need to be studied because that is the future of medicine. So these are some of the reasons why I chose this paper and why I chose to talk about this paper. Now if we look into the strengths and weaknesses, so any even the most perfect paper can have a few weaknesses, and obviously they'll have their own strengths. So when we speak about strengths, one is it investigates a novel approach. So when I speak about novel approach, it's about green nanotechnology because it's very similar to Bhasmikarana process, but a more advanced way, and probably a more easier way when we consider the industrial manufacturing of it. So it investigates a novel approach, and then it employs a multi-pronged approach. So as I was speaking, this I've highlighted a lot of times. So it goes through the entire cycle of preparation of drug, the preclinical studies, and then the clinical studies. So it goes through a multi-pronged approach, and then it demonstrates a promising anti-tumor activity and a safety profile, and it highlights how important it is to be open about research in these concepts. That's also one of the very important strength of this paper. Next, when I come to weaknesses, these were some things which I personally felt as a researcher, it would have been good if they would have discussed these parts or if they would have included these parts. This is a very, very personal opinion. Um, the first one is it doesn't speak much about green nanotechnology. Yeah, of course, green nanotechnology is a very popular field of sub branch of nanotechnology, very emerging branch as well. But for people who are not in the pharmaceutical sector or in the Rashastra and Vaishajakalpana sector, understanding or having a concept or understanding of green nanotechnology is very rare. So the max understanding they'll have is about nanotechnology. So it would have been helpful if there was a brief introduction or a few lines about what exactly is green nanotechnology, because they clearly state even in the title that green nanotechnology and nano Ayurveda medicine. So, when they say that in the title also, it's hard to relate how green nanotechnology can be related to nano Ayurvedic medicine or can be related to Bhasmas. So it'll be easier if they would have given a small introduction about it by saying that, green nanotechnology is where we use phytoconstituents. And in Ayurveda, since Bhasmas are made using phytoconstituents, it's almost a close concept. That's why this paper has been this study has been done considering those concepts. So that is something which I felt was needed there. And again, the second one, the pilot clinical trial sample size was too small. Probably a little bit more number of subjects, because when we speak about breast cancer, triple negative breast carcinoma, it's not that we'll not be able to find subjects for that. So subjects are available. Probably a few more enrollments would have given a better picture about the safety of the drug also. Not even efficacy, but even for safety, this was a very, very small group. And then mechanism of action of NSP remains unclear. So this is something which I felt was lacking, because when we speak about although the paper's secondary objective was to study the efficacy, primary was safety, still some light on pharmacodynamics and pharmacokinetics of the drug would have been very helpful in understanding the safety as well as the efficacy of the product, because that is one thing where even Ayurveda has a lot of issues, because it's hard for us to explain the safety and efficacy of the drugs in terms of pharmacokinetics and pharmacodynamics, how the body takes the drug and what the body does to it and what the drug does to the body. Both the things, it's very hard to explain. So if that was done, then it should have been easier to understand, and it would have been easier to bridge the gap, because bridging the gap and going ahead with the integration is very important when it comes to medicinal approach. Integrating a lot of streams of medicine is very important these days. And for that integration, we need to bridge the gap between the understanding of our science and the modern science. So when I can understand Vata as Vata, it's important that a modern medicine person is made to understand Vata in their terms. So that's where we bridge the gap. And that bridging of gap has not happened here. They haven't explained anything about the mechanism of action of the drug. And then again, long term efficacy is not considered here. So, whatever study has been published is mostly for 6 weeks, 12 weeks. So, the long term efficacy of the drug has not been taken into consideration, and potential long term side effects have also not been taken into consideration. That is something which has to be done. Probably the authors can, publish another study with a long term safety and efficacy part. But yeah, that is something that was also, I felt, a little lacking in this paper. And just to conclude, overall, it was a very good paper in terms of presenting the entire picture rather than going for a single subset. But probably in an effort to present that entire picture, certain things got missed out. So, if those were included, it would have been even better. That's something I've come to conclude about this paper. And with that, I would like to end this. Thank you. Thank you so much for being patient and listening to this one.
Thank you, Dr. Swati. Thank you so much for sharing this such a wonderful paper, especially on Suvarna Bhasma, especially when people, have so many misconceptions about Bhasma. Uh, so this is a really good paper, and I'm so glad you spoke about standardization because that is so important. And that is one of the very challenging things in Ayurveda, which is very difficult to standardize any herbal formulation or any bhasma preparation. So I'm so
Yeah.
glad you spoke about that. One of I have a question that
Mhm.
so this particular study, do you recommend such studies where only one formulation is given to a patient when in reality Ayurved practitioners use multiple formulations, based on various different prakriti, etc., things. So, do you recommend such research studies?
So it's a very, there's no straightforward yes or no answer to this, because as an Ayurveda practitioner, practicing a single herb remedy is very difficult for me as well. Because when we look into the concepts of Ayurveda, even Ayurveda doesn't advocate for a single herb or single medicine study. And along with that, diet, lifestyle also becomes a very huge factor in management of patients when it comes to Ayurveda. But when we speak about that, before we go into clubbing 3 or 4 medicines, it's important that we start by understanding the pharmacodynamics and pharmacokinetics of one single drug. Because when I understand how Swarnabhasma acts in body, then I can go ahead and combine Swarnabhasma probably with Chavanprash or probably with Kushmandavaleha, and then understand how synergistic they are, or how antagonistic they are, or how one balances the other, or how one negates the other. To understand that complex pharmacodynamics and pharmacokinetics, I need to understand the single drug pharmacokinetics and dynamics first. So I would rather say this is something similar to an animal experimentation where we are against cruelty, but there's no other way but to study animals to understand how it reacts in human body. In the same way, we have to start somewhere. So to include a multimodal, personalized approach that Ayurveda does, for example, for a treatment of, say, cancer itself, we do Rasayana Chikitsa, and then we have to go with an aggressive anti-cancer treatment as well. And to understand how all of that works in a patient, we first need to understand how one single drug works. we can, understand probably 2 or 4, 5, 6 drugs which work in cancer, and then we can start clubbing them in patients and understanding how they work on particular prakriti and then vikriti, and all those things. So we have to start somewhere, because the uh, need for empirical research data is very important right now in Ayurveda, considering that Ayurveda is being taken seriously globally. But when someone asks us for a research data, we don't have it. So, although we
Okay.
have anecdotal evidences from practice, and even our practitioners are not publishing much, because if they publish single case studies with multiple drug approach, that can also be a very good way, way in which further research studies can be done with those drugs. But that is also lacking, and we have to start somewhere. So, right now, to begin with, this is something I would encourage, but it's not that on a personal level. Yes, I agree with very personalized individualistic medicine. So I would rather go for a combination study where a lot of drugs are studied together. But to understand that complex interaction, that complex drug interaction, I would rather prefer understanding the single drugs first.
Yeah, maybe like, maybe the next step of the study would be to add more formulations. Because
Yes.
if you study like multiple formulations together, then you wouldn't know which one is actually acting. So
And
maybe like,
especially when we are doing a safety study, we wouldn't know which one is actually antagonistic or which one is harming, which is toxic.
true.
So that's also very important.
Yes, true. I agree with you. So I see we have Pravina with us. Pravina, do you have any questions or do you have any comments on the presentation? You can unmute yourself. Okay. Okay. She's not responding.
Yeah.
Yeah. Anyway, it was a wonderful presentation. Thank you, Dr. Swati. Thank you so much for presenting today. And thank you, everyone, for attending this Journal Club meeting. This meeting will be recorded and uploaded on YouTube soon. Please watch and subscribe to our YouTube channel to stay updated with our latest research in Ayurveda. Please like, comment, and share the videos. Once again, I invite you to become a member and fill out a volunteer form. You can now also subscribe to our Ayur Update newsletter to stay tuned to our projects and events. If you have any questions, feel free to contact us at Ayurveda Research USA at gmail.com. Thank you again. Hope to see you in the next Journal Club meeting. Thank you, Dr. Swati. Bye. Have
Thank
a
you.
good
Thank
day.
you, Dr. Ritika. Yeah. Bye.